Welcome, everyone, to the Washington State Kelp Research and Monitoring Workgroup. This is our January meeting. We're so glad you're all here. We are recording this meeting. Just want to make sure you all know that. And as a quick reminder, we have 3 meetings in 2024, and the meeting dates are on the introduction slide. And in these meetings, the whole purpose of this group is an open forum for information sharing and collaboration. So please let us know what you'd like to hear about. If you want to hear particular topics, if you have some research that you've done, if you have an announcement, please email us because everyone wants to know. We want to share information. So to join or leave the list, you just give us an email at nearshore@dnr.wa. And what I'd like to ask you all to do now is enter into the chat your name and organization and any key updates that you'd like to share. We found this is a great way for people to connect, so we really appreciate it, although it's optional. So what I'm going to do now is pass the, you should all see the agenda in the chat. There's been some slight changes to it. And what I'm going to do now is pass the facilitation on to Bree Turner, who's my co-facilitator in these meetings. And she's going to do the first announcement and then pass it on and facilitate the rest of the meeting. All you, Bree.
Hello, thank you for that. Yeah, so I'm going to be taking off the first announcement. And again, my name is Bree Turner. I am the Marine Vegetation Implementation Lead at DNR with the Habitat Strategic Initiative. And we are very thrilled that we have RFP coming up this spring. We expect it to be hitting the streets in late March, mid to late March. And so wanted to just jump on, don't have all the details yet, but our next meeting, by the time our next meeting will have already been out there. So I just want to put it on your radar. This RFP will be focused on new science, synthesis, and analysis for improved protection and restoration of kelp and eelgrass in Puget Sound. And this will be, I think, very highly relevant to this group. And we're really looking for projects that really advance scientific understanding, critical to informing our response to kelp declines, and really looking for some of that synthesis and analysis of some of the existing data out there. And also interested in projects that are looking at developing a long-term research agenda focused on those knowledge gaps that are really critical to helping gap the kind of science to management piece there. So a little bit about some of the specifics of the funding. The projects will be 2-year awards, and the total investment is up to $2 million. We imagine the awards will be in the $200K to $500K amounts. And that's about all that I can tell you at this point, but again, the RFP will be hitting the streets in March, and we will be having several months where that will be open and ready to go, and hope to see some proposals from this group. So I'm now going to take my update hat off and jump back into the facilitator. Is Betsy on today?
Yes.
We have the kelp forest roadmap as the next update.
Super. Can you hear me all?
Yes.
Great. Thank you, Helen, and all for just creating this forum and being part of this forum. It's such a great group of people. I wanted to give a brief update, and also Helen, she included a link to a kelp forest roadmap on the agenda, which is well worth looking at. This is something that came out of the Global Kelp Forest Challenge that was launched in Tasmania in February of 2023. And there was a big group of people there from all over the world making recommendations about how best to move this work forward. And this particular roadmap captures that work as well as a broader survey to really provide a detailed strategy about how to scale up kelp conservation and recovery. And I think of it as sort of trickle up ecology in the sense that it is drawing from real work that is being done in regions around the world, but really trying to make that available so that those strategies can be implemented across governments, states, provinces, countries, communities. And there are recommendations in there that you can sort of dig into, including stressor management and restoration practices and policy changes and growing awareness. And there's a lot there to chew on, but I want to say that this roadmap really shows that Washington State is doing great work. We're spanning many of these recommendations. There's a ton more to do. But I hope you get a chance to look at the link and forward that as much as possible. Thanks. Was that a question?
Thank
I
you.
didn't quite hear
Yeah.
that.
Just as a reminder, if you're not speaking or you don't have a question, please remember to mute as you hop in here. Was there a quick question? All right. Thank you so much, Betsy, for that update.
Yeah.
And
So yeah, sounds like 3.
Dan, well, there we go. We're gonna move on. Allie with the Northwest Straits. Are you here?
I am. Hi, Bree. Can I share my screen? Oh my God. Let me see if I can. All right. Hey everyone, my name is Allie Simpson. I'm the Ecosystem Project Coordinator for the Northwest Straits Commission. And I just wanted to briefly show off our newly updated story map that is really highlighting the work that marine resources committees have done with kelp monitoring. And so I'm just going to briefly just go through it real quick for you all. We start off with why we're monitoring kelp, why the marine resources committees are involved, and really just an overview of what the project is in general. And then we have a key outcome section, and this section really provides these high level highlights that have come out of the MRC data. So things like the loss of kelp at Meadowdale and Mukilteo, or the increase of kelp at places like Eby's Landing and Possession Point. And so there's quite a few key outcomes in there that you can scroll through that just really put the MRC data in more perspective across the Salish Sea. And then moving on, we have this interactive data map. And this is really pretty cool. I'll give it a second to load here. So each star is a site where MRCs have surveyed kelp either currently or in the past. And when you scroll in, so I'm going to scroll into Shannon Point for a second here. All of these different layers are the annual maximum extent of the survey for whatever year that was surveyed. And you can also click on these layers and get even more information about that survey. So it's pretty fun to just explore and check that out. And then moving on, you notice in the nav, we have a tab for each MRC. And so I'm just going to go to keep with the theme of Skagit County here, go to Skagit Marine Resources Committee. And each of these MRC pages are set up similarly. So they'll have this really great quote from one of the MRC volunteers, a brief overview of the project at the county level itself. And then if you keep scrolling, you'll get a map of all the sites that are surveyed at that MRC. And using the arrow on the side, you'll get a more in-depth description of each of the sites. So just what's happening at the site itself, how the volunteers have described the site, and then a perimeter map showing all of the different surveys that the volunteers have conducted at that site. You can also click on this to blow it up. It just takes a minute to load, so I won't do it now. And if there is a county that has multiple sites that volunteers surveyed, you can keep scrolling through, and each site's can have its own page. And so, yeah, like I said, all of the MRCs are set up pretty similarly with the same type of format. And then at the end, we have a learn more, which is really just sort of showcasing the collective effort of all of the MRCs and what's been happening and how important their data truly is to this work. Followed by just some extra resources related to the kelp monitoring at the county level. And we're really hoping that this isn't just useful for the MRCs, but also really useful for other people involved with the kelp world, just to at least get an idea of all of the work that's happening at the MRC level. And one last thing, I wanted to thank Helen and Julia for all their help with this, because we really couldn't have completed this story map without them. And with that, I will go ahead and share the link for the story map in the chat as well, but it's also on the agenda. And that's it. Thanks.
Thanks, Ali. That's absolutely beautiful and such a great visual representation of all the hard work, that program. We have time for one quick question. Anyone have any questions? All right. Again, congrats. Tom, looks like you're on.
Yeah, I made it. Scurrying around here today. Just a couple of reminders here, and there's some notes in the agenda about web pages. This group has done some amazing work, and there are three opportunities I want to bring to your attention about getting the word out to a larger audience. And the first of those is going to be the Phycological Society of America is holding its annual meeting at University of Washington, end of July, first part of August. It'll be a 3 or 4 day conference. And it's going to be national, international in scope. And it's going to be pretty heavy academic kind of stuff, but this is the kind of stuff, some of this work really could be showcased there. Stay tuned on, but after the 1st of February, there'll be the announcements for call for papers and so forth. Not going to be terribly expensive, but it's not going to be cheap either. I'll just put that out there. The second one is going to be really interesting. It's going to be a year in 2020, over a year from now, in March of 25, and that's the International Seaweed Symposium. And that is truly going to be a worldwide kind of a thing. And a lot of this work would be really good to talk about at that meeting. That one, again, is not for the faint of financially faint of heart, shall we say. The other thing that we're doing, and it's kind of fun, is for years, 24 times, we had what we call the Northwest Algal Seaweed Symposium. And now it includes seagrasses. And that stopped in about 2018, I think was the last one. We're going to resurrect that. And it is basically a local Washington, Oregon, DC oriented, very inexpensive amusement, and to give papers into network and so forth. And we're trying to get that up and going again. And again, that's an opportunity for some of the work that's being done here to be talked about. And certainly kelp is front and center in a lot of the phycological world right now, and take opportunity here to showcase some of that. So just heads up on those. I'll put some more links in the chat for people on that. And if you have any questions, particularly if you want to help and participate in the resurrection, I'm calling it, of the Northwest Algal Symposium, give me a ring or get in touch. Thanks.
Thanks, Tom, for those updates. A lot of great, exciting convening happening later this year and into next. Was there any questions around those upcoming events?
Everybody will be there, right?
It sounds like we'll be organizing a gathering, a happy hour or
So
something.
I have another question, actually, for the communal group, and you put it in the chat. What is the fate of, or what is going on with the Salish Sea Ecosystem Conference. And I just put that out there, and you can respond in the chat. You won't need to talk about it here.
All right. Look forward to hearing what you have to say on that in the chat. Our last update is Cynthia with the Washington Kelp and
Hi.
Eelgrass Health Plan. Yep, there you are. Great.
Hey, morning. My name is Cynthia Harbison with DNR, and I am co-leading the statewide Kelp and Eelgrass Health and Conservation Plan with my colleague Max Showalter, who I believe is also on the call. And we wanted to share with you all our first report from this project, which is our prioritization plan. And that prioritization plan presents a collaboratively developed prioritization framework that's going to guide how we're going to prioritize that at least 10,000 acres of kelp and eelgrass habitat for conservation and recovery actions. Many folks on this call either participated and provided a lot of input through our internal DNR process, through our executive work group, and through attending workshops. So thank you all for all that support and input last year. And we're really excited about applying this framework and beginning to think about and to talk with partners in our 3 pilot subbasins starting this year and over the next few years. So we're going to be focusing our efforts in South Puget Sound, Grace Harbor, and the Eastern Strait of Juan de Fuca starting out to help support that local identification of those priority areas, and then actions and what we're going to do to protect, conserve, or recover those habitats as well. So we're just getting started on this next phase of the work. And we're really excited, and we hope to be working with many of you over the next few years as well. The links are in the agenda, but I can also pop them in the chat as well.
Thanks so much for that update. Exciting. See it moving forward. All right, if folks have any questions for Cynthia, please put them in the chat and check out those linked documents. We are now gonna make a transition into some of our deeper dives. And so our first talk coming up is coming in from Filipe. I think I see you out there. Hello.
Hi.
Hi. You're going to be taking us walk through isolation and low genome diversity in Puget Sound's most threatened bull kelp populations. Does that ring a bell?
Yes, I think so. That's what I prepare for.
Okay, great. So you're going to have about 20 minutes time to present, and then we'd like to have some time for Q&A at the end. And I'll give you a kind of heads up when we're about 5 minutes towards that Q&A, and see how we do. You feel free to share your screen or not.
I think I got it.
Okay.
Right? Can you see it?
It's not in presenter mode yet.
Yeah.
That looks great.
Looks good, right? So thank you so much for the invitation. It's really the best audience possible that I can think for this work. So this is, as always, the result of a lot of students' work. Lily Gierke, a former master's student in particular, led a lot of the work that I'm going to present today. And then I'm going to show the follow-up using population genomic markers as part of a Washington Sea Grant project. There's a lot of partners that I like to thank, probably don't have the time here. Tom Mumford was essential to connect me to all these folks, many folks here in the call today. The partnership with Puget Sound Restoration Fund has been amazing. Washington DNR, the Northeast Straits Commission also helped a lot to establish a network for sampling and to understand more of the system.
This year,
so why do I care about population genetics? So I think it's a way to understand more about the population history of a given species. So things like, where did a certain population came from, how old it is, how connected it is with other populations. All this leaves fingerprints, in the genes that we can inquire, but also at a more of an ecological time scale, we also understand things like the effective population size of a species, the mating system, which might sound a fundamental question, but tells us a lot about extinction probabilities of a species. In today's gene flow, so how likely a species is able to recognize by itself, it's a very important, marine connectivity question. And, very to the topic of today, it gives us a hint of the future also, what is the evolvability and resilience of a species? And, take home message to simplify it a bunch, the more that genetic diversity you have, the more evolvability you have, and the more resilient you should be. So just kind of going back a little bit in this idea of geological time, you probably all know about climate oscillations in geological time, glacial periods, interglacial periods. So this create a strong signature in most species in the temperate systems, both north and southern hemisphere. Essentially, there's this expansion and contraction of the range of the species. So the species will disappear from the colder areas during glacial periods, and it will be restricted to what we call the trailing edge. In the northern hemisphere, that would be the southern part of the range. So the species gets to get old, the populations get to get old in that area. That's what this scheme is trying to show here from Ham and Petit. So with time and age, you get to acquire mutation, which essentially is a generator of genetic diversity. And then during the interglacial, the species is allowed to recolonize back to the north, and you see a decreasing pattern of genetic diversity as you move up north. So there's exceptions to this rule. There's things we call glacial refugia, where the species was able to persist. So there's several of those in the Northeastern Pacific, Alexander Archipelago, Haida Gwaii have been pointed out, and others places that have been pointed out as places where many different taxa was able to stay during the last glacial maximum. Even with the theories about human colonization of Americas are really interesting. The Clovis idea in the last decades has been kind of been debated with a lot of evidence that humans might have been in the Americas for a much longer time. And the very interesting hypothesis to us is this idea of the Pacific coastal route, or also known as the Kelp highway hypothesis, that would allow humans, based on the ecosystem, the kelp forest ecosystem, to move coastally into the Americas. So if we look, just kind of like going back a little bit to the work we did in giant kelp, and we think about the patterns I just showed. So this bar plot here just shows within population diversity, and you can see from south to north giant kelp as this decreasing within population diversity, which is evidence that during the last glacial maximum, giant kelp was not present in the northern parts of its range in the northern hemisphere. And also shows that the south is really the hotspot of genetic diversity. So really fits this model, and probably was not giant kelp that supported the kelp highway hypothesis. If we move to some of the topic today, so bulk kelp, we just published the work of Lily Gierk, the first kind of study across the whole distribution range of bulk kelp, and we don't observe the same pattern as we did for giant kelp. So here we observe that yes, we have in the south the hotspot of diversity as expected, so populations that are old, California and Oregon, but also in the north, from Haida Gwaii up, we see this high diversity. Here we have a allelic diversity on the left side of these bar plots, and on the right side we have private allele, which is a really good evidence for refugia. So this is the Alexander Archipelago sample, really supports that the species might have stayed here, which is very interesting. In Washington, we see that we have lower diversity overall, and we have this solid brick color, which is the inner Washington, so the Salish Sea, Strait of Georgia, and Puget Sound, which is the lowest, where we find the lowest values of diversity. So it might have been that, if I can speculate, that bulk kelp might have been important for a possible kelp highway hypothesis for humans. We know that the Salish Sea was only colonized about 16 to 14,000 years back during after the last glacial maximum. So we know that the kelp that lives there is relatively new. And this was kind of evidence on the data too. This network tries to show genetic differentiation between sites. And we see that California, Oregon, and the Alaska Hawaii group, they still share a lot of ancestral, what we think are
Hi,
ancestral
Kaylee.
alleles. And then, so we see here on this histograms here, these green colors, when we force the method to show us only 4 groups, that there's still some common ancestral alleles between the southern and the northernmost group. So I'm not going to explain to you the kelp life cycle, you probably know, but there's a lot of interesting things that came out of this study relating to to open more other doors, as always, of research in particular the mating system of Nerio. So we saw both this rare combination where we saw evidence for selfing, and that's the reproduction between male and female coming from the same sporophyte. The species has what we call a mixed mating system, both self and outcross. We saw evidence for that within each population, but we also saw these large swaths of coast where there was very little genetic differentiation, which is evidence for long dispersal and long distance gene flow. So it's been pointed out before that the presence and the way that the spores are released from bulk kelp might actually allow for this mixed strategy, which is really interesting. So it seems that this study kind of suggested that bulk kelp might lay in between Pastelsia, with all the work that Kathy students have done before and showing no inbreeding depression, reliance on selfing, and the other way, giant kelp. So these are the 2 closest species, and giant kelp, which shows inbreeding depression and outcrossing evidence. So bulk kelp might be more flexible in a way. So we all know that bulk kelp has been declining in California in many places. Also, I borrowed these images from DNR. We all are very aware of the status of kelp in the southern Puget Sound. This is an area that we've been, the whole Puget Sound, we've been working with the Puget Sound Restoration Fund to create a biobank, and thinking about understanding more the genetics here and the possible restoration using the biobank or other techniques. My lab is invested in biobanks. We have very large biobanks working with Puget Sound, working with other partners in California for both kelp and giant kelp. This allows for continuous access to seeds. There's a lot of other fundamental questions that we still need to understand about biobanks, but it's very promising. So recently, as part of a Washington C grant, we moved on to markers across the entire genome, which we call population genomics, to understand this system. And we're also running work, the same type of work in California. So if I zoom in into what Lily found in the Puget Sound using microsatellite markers, this was only 7 microsatellite markers. We see that in this area, there was the different colors here in these pie charts, dominant colors, show groups of different genetic differentiation. So we have an outside group and an inner group. And in this new plot here, what I show in the x-axis is kind of like the effect of isolation in the Puget Sound. This distance is being measured from 0B Tatoosh Island, and then I'm measuring distance from, the entry of the system into here at the bottom. The sites are organized in the y-axis according to latitude. So this would be your southern Puget Sound, and this would be the sites in the northern part of the Strait of Georgia. And the diameter of these circles is just within population diversity or allelic richness. So we see that as a decreasing within population diversity, not so much in the Strait of Georgia, because that's not really a closed system. So that makes sense. We also observed in Lily's study that we could explain a large percentage of the genetic differentiation using just simple oceanographic connectivity. And we work with Tarang from PNL to get models of circulation for the system. So we have evidence that yes, they're being connected by currents, and there's an effect of connectivity on the levels of diversity. So this was kind of like what we had before, and then we got funded by Washington C grant to go a little bit deeper here and to just essentially provide more information to understand if we can use a genetic rescue to lay out a hand to bulk kelp in the system. So that, if these two groups that I showed are actually locally adapted, we might have a situation where we have to think carefully about employing genetic rescue. So the whole goal of the project is really trying to understand this question: is the Puget Sound kelp locally adapted or not? And to this, we have like a three-pronged approach to tackle the question. So what I'm going to talk to you, and it's just like the first results, is the first idea is to identify genomic signatures of selection through whole genome sequencing. Then we're also going to, using the gametophytes, we're going to look at gene expression using experiments where we expose gametophytes to different temperatures. And then the golden rule for local adaptation is still a reciprocal transplant, which is being deployed this spring by the Puget Sound Restoration Fund using gametophytes from the biobank. So today I'm going to be mostly talking about the first results on that point one, so whole genome sequencing of bulk kelp gametophytes. So we actually used haploids, 100 haploids from the gametophyte bank. Here you can see where we got them from. So pretty much the Straits and all the Puget Sound. So we pre-treated these cultures with antibiotics to get more of the sequences being actually kelp and not bacteria. We target for 20 times coverage and eventually got 12%. So you still get a lot of bacteria and other contaminants. Deep dive into the scripting and bioinformatics. I'm not going to spend too much time explaining, but we have a draft genome for bulk kelp as collaboration with the California Conservation Genomics Project. So this is really great because we can map the sequences from all these gametophytes into actually a genome, understand the distance for each variant to the other one, and eventually get into functional genomics when we learn more about these genomes. The Joint Genome Institute is currently working with us to annotate this genome, which is great. So we end up with about 700,000 SNP markers, so some orders of magnitude more than the microsatellites, which is great. We run some admixture analysis to get at the level of genetic differentiation in the system and contrast that with what we've learned from microsatellites. We were able to estimate linkage disequilibrium because all these variants were mapped against the genome. We run a marker environment association analysis. This is just the first of a very large number of analyses that we have to do around this topic using an RDA in R. And then we kind of used, we didn't add a mutation, so we did our best to use the giant kelp annotation to learn more about some genes that might be associated with different environmental variables. So jumping ahead to the results. So here I'm showing, again, this idea of genetic differentiation plotted as percentage of the individual that belongs to a certain group of genetic co-ancestry, if you will. Think about, 23andMe for kelps. Right, so here, this solution is the solution for two ancestral groups, and you see that it's not that far from what we've learned from microsatellites, so the southern sound separated from the rest. But, because we have so many more markers, we actually can go into solutions with more groups and understand more of the substructure. So this would be the solution for 3 groups, and this is the solution for groups. And beyond this, we start seeing a lot of admixture between the groups, which for me is a sign that we're trying to dig too deep from what the markers are telling us. So we found these 4 groups, so more information and more subdivision than we got from microsatellites was expected, given that we have orders of magnitude more markers here. Now this is probably one of the most interesting slides in the talk, in my opinion. This is now looking at the within population diversity, is heterozygosity. So how many of these variants, how many of these single nucleotide polymorphisms were heterozygous, right? So if a population is diverse, you're going to have a high level of heterozygosity. If you have low heterozygosity, you don't have a lot of diversity. So when we, on this bar plot, we see heterozygosity, and if we go all the way to Squatian Island and some of the sites that are more affected, we have heterozygosity below 5%, about 3%. So extremely reduced. It's almost like full homozygosity across the system. And the density of this panel is really, really large. So this is very concerning for the diversity of the kelps in that region, in the fate of those populations. So this was also obvious from the linkage disequilibrium plot. This is linkage decay. You all probably heard about crossing over in linkage. So we can see a normal pattern of linkage on 3 of these groups, and then the inner group as what's expected when you have high homozygosity, where you have, it really doesn't matter what type of crossing over you're doing, you're always shuffling the same genes because there's not much cards to shuffle. So then eventually we did this association study, having all these SNPs with different environmental variables. You know, and this is very preliminary, but the idea just to show you a little bit of what the capacity of the methods that will allow us to pursue this association with environmental variables, and then point out specific regions. The red dots here, those are variants that are associated in this case here with sea surface summer temperature, up here, you know, water, light penetration in the water column, chlorophyll in the winter, salinity here. So we have a plot for, you know, associations with salinity here and other variables. And eventually we can summarize all this and explore the associations of different environmental factors with different SNPs. There's a lot of challenges doing this analysis, a lot of false positives. So the idea is always to do a lot of redundant analysis in different methods and try to get the redundant evidence. And then eventually you can dig in and get to these SNPs as we learn more about the annotation, try to understand if there is any functional genomics that makes sense, and then match that with the gene expression work that is also going to be part of this project. So it's just a final slide as a conclusion. So the genomic analysis, which is very preliminary at this stage, we are in the process of hiring a postdoc that's going to help us flesh out this. Confirms that the very threatened status of bull kelp in the inner Puget Sound. I think this opens the door for the debate if it is even worthwhile trying to preserve that genetic, the genetic material that is there, given the high homozygosity. So a very conservative genetic rescue measure would be to source restoration efforts with a close to higher genetic diversity pool. So Seattle, although the kelp beds aren't that large, is still considerably much higher heterozygosity than the southern South. So that could be a possible source. So we see this increased demographic bottleneck associated with distance, but again, the severity is limited just to the innermost sites. And we have already many candidate gene loci as part of the first kind of catalog of genes that might be associated with some environmental factors. The analysis becomes very complex, including Squashing Island, because of that high homozygosity that creates a lot of confounding signatures in the data. So we might have to do this between Seattle and the Strait of Juan de Fuca to get a really sense of our question if there is any local adaptation. And we're very happy with the high density SNP panel for the species. This will allow a lot of progress in the next decades to come in terms of functional analysis, possibly breeding of bull kelp using these new genomic tools. Thank you. Take your questions now.
Thank you. That is just amazing. I am sure there's going to be lots of questions. And as a reminder, you can put your questions directly into the chat, or you should be able to have the raise hand function, and I can call on you as we go along here. And it looks like Tom has a hand raised. Question.
A couple of things to think about here. One is there a possibility for going back and looking at older herbarium sheets and those sorts of things from South Puget Sound to see how this may have changed at different sites over the last 50 years or so? In other words, can you get some of this genetic information out of older specimens? And would that tell you anything about what maybe happened in the last, since the serious decline?
Yeah, you just made the same question to Marlene Pinsky 2 days ago, Tom, and or 1 day ago. Yeah, and he's way more optimistic than I am. I think for kelps are already recalcitrant, so we were so happy that we got this quality data from fresh material. Marlene thinks that he can. Again, he's just optimistic, so he will tell you, yes, I feel that I might be able to do it. He's just arriving to the world of kelps too. I'm very skeptical of being able to use genomic tools where you need high quality DNA And you can use things like forensic techniques and to get specific regions. So one day, say if we find that there's a certain gene that seems to be really associated with a possible local adaptation, then we might be able to push that particular region from that specific region from an herbarium, but not perhaps the whole scan of the entirety.
Right. A second point that I brought this up yesterday, we were in this discussion with the, what was it? EPAC, whatever. Are we trying to restore the wrong phase of this kelp? Should we be, and I pull this out not necessarily for you, but everybody, should we be looking at restoring the gametophytes, not the sporophytes? And is there any reason for that or not? 'Cause all the sporophytes, near as I can tell, the gametophytes are all still in the lab somewhere.
Yeah, but sorry, your question is again, Tom, should we be
So
what?
are we restoring the wrong thing? I.e. thing being the phase of the life in the life history.
I don't see them as disconnected. I think if you store the gametophytes, it is a great question in what, for me, after working with these biobanks for 5 years, there's a lot of questions that have opened, right? So that I think no one is really answering. And those have to do with what is really the fate of the sporophytes that you can produce out of gametophytes as they age in your collection, right? I think there's a lot of unanswered questions that from what I've learned in the last 5 years. And because it's much easier to store gametophytes. They're theoretically perpetual, right? You can't do that with sporophytes. But the promise is that you can build that sporophyte out of the same male and female gametophyte in 1 year, 2 years, 10 years, 20 years. And what I say is that, but can you? What are the epigenetic effects of that process, right? No one's looked at that, right? So always, right, you can build clones, essentially. You can build the same clone. So by building a clone, you can do absolutely control experiments where you have the same exact genotype. So you could actually study this. You just need to get the funds, right? You can understand are you affecting the fitness, and if so, of the same exact clone as it is produced by gametophytes that are stored 1 year, 5 years, 10 years, 20 years, right? And are you seeing any, the effects of somatic mutations or epigenetics? Is there recovery? Because, in the end, resources are going to be limiting. And I think the way that we know now is either biobank as we know them now, or perhaps, the cryopreservation, if we get really to the bottom of that and we can use it. But again, the same question will be asked, at least in my mind, even if we cryopreserve them, what is the fate, what is the effect on the fitness of that sporophyte that you produce as the gametophytes age? And that question is not that easy to answer because you pretty much you need a common garden for kelps, right? The ocean might not be the best place to test this because the ocean every year, every season is different.
Looks like Helen has a question for you as well.
I do. Thank you so much, Filipe, for this. This is such important work. I'm hoping to go local. So I'm hoping you'll go back to the slide that looked at heterogynous and it showed the real low amounts. There we go. So I look at this slide, and many others may also look at this slide. And I'll focus on South Puget Sound because it's the most straightforward. And I look at these really low numbers, and I compare them to what we know about at least in recent years, the stability of these populations. So we know that 19 squoxin has been more persistent than other sites in South Puget Sound and really declined recently. 18 Day Island and 17 Salmon Beach. 17 Salmon Beach is quite stable, and it's in the most connected portion, meaning that there's bull kelp all along the narrows. And so there are populations it's a much larger population, whereas, of course, Squaxin 19 is very isolated at this point. And Day Island also looks relatively healthy to us. We know less about it. So I guess to think a little bigger, when I look at these three really low genetic diversity sites, I think that some of them are stable, and at least in the way we know it. And so I start wondering about the environmental conditions and also connectivity with other, just the size of the nearby bull kelp population. And I'm wondering, Filipe or others here who know a lot more about genetics than I do, how do you think about the stability versus decline in light of those numbers, those genetics numbers?
Wow, that's really interesting, Ellen. So, you know, this data has the promise to allow us to date some of these declines, but the decline is so big in terms of homozygosity that, again, there's confounding forces there. It's going to be a little bit challenging, but there's the promise is that we can try to understand, can try to get a date for when this decline happened, which is going to be really interesting out of this data. So there's demographic analysis. Since you have so many markers, there are demographic methods to try to age when this decline happened. That's going to be really interesting. That's really interesting that site 17 and 18, so the island and SB, they seem to be still large and connected. Tarang has new models that we can try to employ here to understand more the connectivity in this system and see what explains. I know you guys are been working in the cover, in the historical cover. So I haven't shown any of that because I haven't touched any of that. I would love to collaborate to actually include that layer. You know, the habitat connectivity is something that we've done a lot in California with giant kelp. You know, the idea there is, you know, each patch has its size and it's a stepping stone of genes to the others. So you have the ocean currents that Tarang would bring as the force that disperses, and then you have the size of the patch, the historical size of the patches as the size of the source of the kelp beds, right? So putting all those things together, you can try to understand why that decline happened there. It does seem that you are puzzled about some of those, and I'm too. That's very, very interesting. It's almost like hints that it could be a little bit more older than something that just happened recently. The fact that SB is already showing that decline. I mean, it is kind of cool that that pattern, the decreasing pattern, is still there. It's like a stairway, right? You're always coming down as we move into the further in the Puget Sound. But then it gets really bad by the time we get to site 16.
Thank you for that answer. We're starting to get some questions coming in through the chat, and we have one from Wendell. The question is, do these recent data suggest evidence of selfing of narrow in South Puget Sound?
Yeah, you know, it actually, it doesn't really matter because the genes are essentially the same. So even if say like in Squashing Island or Day Island with this many SNPs, it even doesn't matter what mechanically kelp is doing. So we know that it's probably selfing more just because the population is smaller. But even if the population would be larger, which is not, the genetic consequence would be the same because the genes are the same. So it doesn't even matter. It's like, normally I say it's just like my dog. I have a greyhound and my wife did the genetics of the dog, and it turns out that the dog has half sibs and full sibs all over the world because this breed was so selected that they all share the same genes. So whatever negative consequences there are or there aren't for selfing in bull kelp, they're there. Doesn't matter if it's outcrossing or selfing in those sites. The study by Gierke suggested that, yes, there's a good amount of selfing occurring at most sites, but at the same time that they can spread genes far, which is really interesting. I don't know if I answer your question, but
Yeah. Wendell, feel free to chime in if that hit the mark.
Yeah, thanks. That was great. And yeah, great talk.
There was just a kind of quick logistical question actually on this slide. Was talking about number 14. Did you see it was Port Townsend? It looks like it's down on like Marrowstone Island area, or Indian Island rather. Is that should it be north of I was just wondering if that was should it be north of Port Townsend?
I would have to check that.
Okay.
Yeah, that's a great point. I have to check that. Yeah, that might be wrong there. I know for sure that for the microsatellite site was in Port Townsend, and
Right.
here this is either an error, graphical on the plots, or it was the closest sample we got from there, and we call it the same name to have.
Okay.
Just because we got so accustomed to Port Townsend area, so I'll have to check that. Thank you.
Okay, thank you.
Not all these sites are exactly the same. We used for the Lily Gierke study. So
Great. Looks like we probably have time for one last question, and there's just loads of stuff coming in the chat, so you can feel free
Yeah, I'll jump in because I'd love to take advantage of some people we have here today.
Sure.
To answer a question, which is Celine asked a question that PSRF is set to launch a reciprocal transplant experiment mixing kelp from the Strait of Juan de Fuca with Central Puget Sound. And I was hoping Filipe, PSRF, the group could sort of talk about what I was hearing from Filipe's talk was that Central Puget Sound might be a good place, one of the more diverse areas. And so what would be the goal of mixing Eastern Straits with Central Puget Sound? So, and this is really moving into how we might use these findings for research.
yeah, so the
other tests.
So the reciprocal transplant goal is really, so the goal is really trying to understand if there's localized adaptation in these two. The project was written when we only had the microsatellite data. So essentially we're considering this as two groups, a lower diversity group and a higher diversity group. And we now know more how that pans out. But you can see that, this Seattle region is still, it's the colors on the bar plot here, right? So they're still differentiated from the strait, and the strait is still richer than even Seattle. The question remains, if we want to understand if there's any sort of adaptation from the organisms that live within the sound to their local environment, and perhaps even the ones in the strait to their local environment. And still the best tool to do that is a reciprocal transplant. So where you just use some of the material from environment 1, put them on environment 2. You also use the same protocols, put environment 1 back in environment 1, and you do the, vice versa. We had a third level to that, which I think it's always an interesting level, which is we're actually crossing them experimental too, to see if there's some evidence of hybrid vigor in that cross. So we'll have 3 responses. We'll have the classical reciprocal and the hybrid vigor hypothesis. The other avenue, so that's the big avenue to try to get at reciprocal, at local adaptation. And then then I mentioned today that in terms of understanding true genomic signatures of selection, we might have used Seattle to answer to that question because of all the confounding signatures that such a low heterozygosity in, say, Squashing Islands, brings to just makes the whole challenge much difficult. I guess the idea there, my conservative idea there, is that if we have local adaptation to the Puget Sound, I think it's reasonable to think that we would have it in Seattle already. But we'll try to extract what we can from Squashing Islands.
Thank you so much. The questions are continuing to flow in the chat. And so if you can or have time to respond to some of those, unfortunately, we're going to have to move on. Thank you so much. This is just absolutely fascinating research and really has a lot of implications, as Helen noted, for kind of moving the needle forward. So thank you again so much. And definitely take a look at the chat for some great questions, some additional great questions we unfortunately don't have time for, or maybe we can loop back around at the end of the roundtable. All right, so we are moving on, and our next presenter is Varoon, who I've just met this morning. There you are, hello, who's tuning in from the University of British Columbia and will be talking about a recent publication, Kelps on Demand, Closed System Protocols for Culturing Large Bull Kelp Sporophytes for Research and Restoration. Welcome, Varoon, and please feel free to share your slide.
Okay, can people see that?
Yeah, looks great.
All right, well, let me know when I can get started.
Go for it.
Okay, well, thank you so much for the introduction. Yeah, so as Bree was just saying, my name is Varoon. I'm a PhD student in the Martone lab at the University of British Columbia. And today I'd like to present on some work that we've been doing on developing ways of growing bull kelp sporophytes in a closed system culture for research and restoration purposes. So I'm guessing everyone here is probably familiar with bull kelp versus Leucaena. This is one of the few kelp species that is capable of forming canopy forests that come all the way up to the water surface. And in some areas of its range, this is the like the Strait of Georgia and Puget Sound. This is the only canopy forming species of note. Unfortunately, and again, as I'm sure everyone here is aware of, there have been extensive declines in bull kelp documented in many areas of its range. This includes declines in California, in Washington, notably Puget Sound, as previously mentioned, and also in British Columbia. And in response to these extensive declines of bull kelp in response to a variety of environmental stressors, and also of other important kelp species, in recent years, there has been a substantial increase in interest in kelp research and restoration. So here I'm just showing just a few representative groups just off the top of my head who I know are working on kelp, but really this is just a tiny, tiny handful out of all the different initiatives and projects and organizations that are currently active in the kelp world. And so many of these initiatives and projects will probably also involve cultivating kelp. However, a lot of the more established kelp cultivation techniques tend to be more geared towards kelp farming for commercial purposes. And because of this, they may or may not be applicable for research and restoration. And this is especially the case for the cultivation of kelp sporophytes, which are the macroscopic life stage that most people will tend to associate with the word kelp. So traditionally speaking, when we think of kelp cultivation, this involves outplanting in ocean-based farms. And these farms can face environmental or legal restrictions on where they can be sited. The farms also tend to often, but not always, operate on relatively large scales. And there's this inherently limited ability to control or monitor the exposed culture environment. On the other hand, research and restoration may require cultivation of kelps away from the sea. For example, at an inland institution that wishes to participate in kelp research. It may require cultivation at smaller scales, for instance, as new methodologies are being tested out, or under very tightly controlled conditions for research. And so these were all considerations that we had as we aimed to expand our kelp research in the Martone lab at the University of British Columbia. So UBC, unfortunately, is not situated particularly closely to any suitable kelp farming sites in the ocean. UBC also does not have any specialized flow-through seawater infrastructure, which I know other places have successfully used to grow kelps on land. And because of these two limitations, we decided that we would try to grow bull kelp sporophytes in a land-based closed system culture with the goal of being able to produce bull kelp sporophytes all year round under controlled conditions for ecophysiological, developmental, and applied research. And so before I keep going in this presentation, I thought I'd quickly mention again that our methods have been published in the Journal of Ecology as an open access article. So if anyone is interested in the specifics of what we did, our methods should now be available to everyone. So getting started on the methods, well, before we were able to grow any kelp sporophytes, we first had to start by growing some gametophytes. And so this portion was done following established protocols that are more or less universal for all kelp species. So we first collected sori from wild-growing bull kelp. We cleaned these and then induced them to release spores in the lab. We let the spores germinate and develop into microscopic gametophytes, which we maintain under red light. The red light basically just keeps these gametophytes in a permanent state of vegetative growth and just inhibits their reproduction. And then whenever we decided that we wanted to produce sporophytes, we would then take these gametophytes and then expose them to white light and water motion to get them to make sporophytes. And so at this stage, this is where the young kelp would typically get planted out into the ocean. But instead, we just kept growing them on in the lab. And we realized very quickly that bull kelp can be a pretty challenging species to keep alive, especially without the luxury of having a flow-through seawater system. It is a very sensitive species to many environmental conditions, and if it's not happy, it can die very quickly. However, of course, with the massive amount of interest currently in the species, we decided to persevere, and eventually we were able to determine the specific conditions that are required to keep bull kelp alive and thriving in a land-based closed system culture. And so these included, for instance, having a relatively low stocking density, having a lot of water motion in the culture, having balanced nutrient levels that were neither too high nor too low, and also replacing a culture medium on a regular basis. Culture medium in our case being natural seawater that was brought up to UBC in a tanker truck and then stored in a reservoir before use. And so we had two main approaches that we used to grow bull kelp in the lab. The first one that I'll talk about is growing them in tumble culture. So in tumble culture, the kelp are just freely floating in the culture medium and not really attached to anything. This is, of course, perhaps a bit less relevant for restoration outplanting, but it does make the kelp much easier to handle for, say, research. And yeah, so in the tumble culture, everything is just constantly kept in motion by air that is pumped into the water. And here we took kelp gametophytes and we inoculated them into these smaller 26-liter tumble culture vessels. And we just let them do their thing and develop sporophytes and just let the sporophytes grow. And once the sporophytes were large enough at around 5 centimeters or so, then we'd take them, we'd start straining out these smaller tumble cultures and removing only the largest sporophytes, then put in these bigger 600-liter tumble culture tanks. And in these larger tanks, we were able to get the kelps to grow to around 40 centimeters long. At that length, what happened was that the pneumatocysts became so buoyant that the kelp stopped tumbling, and we then had to take them out. Fortunately for us, 40 centimeters was already more than enough for our research purposes, at least for the current time being. One more interesting thing that I'd like to mention about these tumble cultures was that since we only kept straining out the biggest sporophytes every week, this left behind the smaller gametophyte fragments and smaller sporophytes, and these could then keep growing and produce the next crop of sporophytes. And so in this way, we have been able to get these smaller tumble cultures to produce crop after crop on a weekly basis of little baby bull kelp sporophytes for more than 6 months on end after only an initial single seeding with gametified fragments. And the other way that we grew bull kelp in the lab was growing them attached, which is a more natural way for bull kelp to grow and potentially more relevant for restoration and outplanting. And so in this case, we took our gametified fragments and we seeded them onto these ceramic tiles, on which we then allowed them to reattach and then produce sporophytes. And once they had produced sporophytes, we then transferred these tiles into our custom seaweed growth flumes. And so just a word of explanation on the growth flumes. There's one shown on the top here. They're basically just these little recirculating raceways that were designed and constructed specifically with seaweed research in mind. So their main feature of note is that they can be programmed to produce these really complex oscillating patterns of water flow to basically just mimic the natural marine environment. And as the bull kelp sporophytes kept growing up in these flumes, we had to keep thinning out the larger sporophytes so that they didn't get overcrowded. And what we observed was that every time we thinned out the big sporophytes, the remaining gametophytes on the tiles would then respond by producing a new flush of smaller sporophytes, as you can see kind of in this image in the lower right. And so similarly with the smaller tumble cultures, we were able to get tiles to just produce crop after crop of little sporophytes for more than 6 months on end after only seeding them one first time. And so bull kelp is a very notoriously large growing and fast growing species. And so even with the thinning, the sporophytes very soon started to outgrow this limited space in the flumes. And what we then did to allow them to keep growing was we actually made these tiny little harnesses out of just conventional aquarium airline tubing. And then we tied the nematocysts over to one side of the tank. And so as the blades kept growing, then we just kept trimming off the ends. And as the stipe kept elongating, we just kept coiling it up into this tight coil at the bottom. And in this way, we have been able to get bull kelp to grow to be around 6 meters long in a tank that was about 1 meter in length. And so other seaweed species have successfully been grown in a closed system culture, including for commercial purposes. But at least to the best of our knowledge, this is the first time this has been attempted with bull kelp. And so because of this, I did want to quickly highlight some of the potential broader implications of closed system cultures, specifically as they pertain to kelp research and restoration. So one key feature of a closed system culture is that culture conditions can be very precisely manipulated. And so this could be a big asset for research, in particular for ecophysiological research, studying effects of various environmental stressors on kelp over longer timeframes. And being able to really precisely manipulate culture conditions could also enable year-round production and research on kelp instead of relying on a more limited natural field season. Another important feature of these closed system cultures is that they tend to be relatively self-contained. And so because of them being because of this quality, this could enable kelp production and research potentially quite far inland without relying on access to suitable kelp farming sites in the ocean or access on specialized and coastal flow-through infrastructure. And being relatively self-contained could also enable improved biosecurity, not only in the sense of pathogens or pests that could attack cultivated kelp, but also in the sense of preventing cultivated kelp from escaping into the environment, which even if we're working exclusively with native species, this could still pose a concern of introducing local genotypes in areas where they don't belong, as we currently are still, figuring out how different populations are connected. And so overall, we do think that closed system cultures could be potentially a useful tool that could complement existing research methodologies and just increase the scope of what kelp researchers are capable of doing. And so just using the Martone lab as an example, so having this consistent, accessible year-round supply of kelp sporophytes has definitely been a boost to our kelp research. We've been able to conduct detailed observations and experiments on bull kelp morphogenesis and development. We've been able to conduct plasticity experiments, looking at how bull kelp responds to different flow conditions. We've been able to send off healthy and growing kelp tissue to a different lab at UBC for hormone analysis in the middle of winter. And we also have future plans to do nutrient uptake experiments, long-term thermal physiology experiments, microbiome analysis and manipulation in collaboration with the Parfrey Lab, from whom you'll be hearing a bit more of later today. Parasite infection trials with PhD in collaboration with a PhD student from the Gary Luck Lab at UVic, and also strange lectin experiments. And one more thing that our closed system cultures have allowed us to do is actually also provide supply young kelp in support of a local restoration initiatives in the Vancouver Harbor. So in this case, we are basically just growing little baby bull kelp on these ceramic tiles until they're large enough to survive challenging environmental conditions out in the field. And then we are handing them over to our restoration partners who are currently trialing out different ways of outplanting bull kelp. And we are continuously improving on our methods with a focus on increasing our resource and labor efficiency. Currently, our main goal is to further reduce our reliance on natural seawater by recycling our culture medium through UV or mechanical filtration. And we are also working on growing even bigger kelp in our systems, with the eventual goal of being able to close the bull kelp life cycle in the lab in a closed system. So in conclusion, we found that it is indeed possible to grow bull kelp sporophytes in a closed system, potentially to quite large sizes all year round with multiple approaches. And we think that closed system culture of kelp could compensate for some limitations of more established culture approaches in research and restoration perspective, and thereby increasing possibilities for kelp science and conservation. Thank you very much. And that concludes my presentation.
Thank you so much. Really fascinating project, and congratulations on your success there. We are starting to get some questions coming in to the chat, but also folks can feel free to raise their hands. I will start with the first question I see in the chat. And I think you addressed this, but did you notice any difference in the morphology of the juvenile sporophytes after you scraped them from the slides and they started growing in the tumble culture vessels?
Yes, we actually did. And this actually is mentioned in the publication itself. But yeah, I thought it was a relatively small detail. So the freely tumbling kelps, what they tended to look like was they had quite broad blades, big nematocysts, and relatively short stipes. On the other hand, the kelps that we grew attached, what happened was they tended to grow quite long. So they had really relatively small nematocysts for their size, quite narrow, really long stipes. So there definitely seems to be, they definitely are very plastic in response to, I guess, different mechanosensory aspects of kind of what they're sensing in their environment. And as actually we let the kelps grow larger and we started kind of like tying them up and coiling up the stipes, what we also noticed was that for these really big phalli, they had really underdeveloped holdfasts compared to kelp from the wild of a similar size, just because presumably once we started coiling up the stipes, there wasn't really any tension acting on the holdfast anymore. So yeah, there's a ton of interesting plasticity that we observed, which would be a great avenue of future research for someone.
Tom, I saw you had a couple questions in the chat, but you got your hands up, so I'll just give you the mic here.
Yeah, this is really great stuff. I find this really, really great. And it opens up so many possibilities for all kinds of different experiments. So thank you for doing all this kind of a thing. Interesting. Did you ever go back and look at that old paper by Mary Jo Duncan, the red light, blue light morphology business, and whether or not did you play at all with light quality in any of these growth experiments, or could you that might influence some of these morphologies?
So we haven't really done that with different light wavelengths. We've cultivated all of our sporophytes just with white light. So that definitely would be a very interesting thing to try in the future. The one thing we did do was actually try varying light intensities to see if that might potentially slow down this rampant stipe growth that, in our culture system at least, was not really something we were immediately desiring. And we honestly did not really notice any slowdown in growth, even as we really ramped up light intensities, even to the point where the irradiance was actually bleaching the kelp. So they just still they didn't really seem to change much in their growth behavior. But as far as like exposing of different wavelengths, I do agree that that would be very interesting to try in the future.
I recall the story was that there was something about at depth, you don't get any red light and you get the stipe elongation. And when it reaches the surface, the meristem and the stipe switches to the meristem and the blade and with the red light then. And so the stipe stops elongating, the blades elongate. So there's this, it'd be fun that'd be a great opportunity to play with this.
Yeah, I do agree. Yeah, I do recall reading that publication some years ago. I do think, I think that could be a very interesting area to look at.
Old timers, yeah, from back in the day. Neat, good work.
Thank you.
Filipe, I see your hand up.
Yeah, I can't stop looking at this system. It's really fascinating. There's something you said near the end, which kind of ring the bell, the closing the system. I think closing the system, reproducing these kelps, sporophyte, gametophyte, sporophyte at will, it's really in the agenda. I think, there's folks doing it, but, is kind of like the future, I think, for breeding, for aquaculture, for fundamental research. Being able to complete the cycle, it will be amazing. You probably know this, but it might be that for fast cycles, the way to go ex situ might not be your system mimics to the best possible way the organism as it is found in the wild. But for a cross cycle to get, in this case, to get the spores out of the sporophyte in the lab, the fastest system might mimic the full development of the individual, might be something else. For example, you might know the way that Mike Graham does this with bull kelp, right? If you don't, I'm not going to tell you, but you probably know, something to think about, right? That it might be that for closing the cycle, it might not be mimicking the organism. It might be something else that brings more efficiency.
Yeah, no, I completely agree with you, especially with how large and unwieldy bull kelp can get. Yeah, I had a lovely chat with Bennett Bugby at WSN last November. And so we definitely exchanged some ideas about, growing bull kelp in like just closing life cycle. So I know that they've been doing this with a flow through seawater system on land for some time. And so we have tried something similar by just like crowning what they called just like crowning the kelp and just having this floating cap with the blades. And what we found was that in our system, the sporophyte thalli tended to rot faster than they could continue development. I believe personally that that's simply because in our closed system, there's a much higher microbial loading of things that are potentially degrading the kelp too. And so with their methods, we weren't able to close the life cycle. However, I have also separately made the observation that if you make this important cut and remove the rest of the stipe and the nematocyst, before they've actually formed this internal gas cavity, that the wound actually fully seals over. So you might potentially be able to get something where that heals and then continues developing without this open wound. So it's definitely something that we want to try out in the future. And something we also thought about is actually, instead of doing this, just potentially playing with photoperiods or water chemistry in a way that induces premature source formation on a relatively small thallus. But yeah, that's just something that we're working on. We haven't
Awesome.
really done it yet.
Yeah, that's the new frontier. Yeah.
So I'm going to jump in. I think Bree's connection got limited, and ask a question that I just posed in the chat, which is a general question for people like me who don't do bull kelp culture, which is, I think this is to Varoon and all. How common are closed culture systems in Salish Sea right now? And are they really important for people who are doing this work from a permitting, research, or biosecurity perspective? Or are most people using open systems? And by the way, Varoon, you're getting lots of other comments in the chat too.
Okay, yeah, I'll try to look through them after the question period. So I can't answer this with a ton of confidence. What I can say is that, to my knowledge at least, and to Patrick Martone's knowledge, I'm not aware of anyone else getting bull kelp in a closed system. If there is, definitely please speak up, and I definitely love to chat and exchange methods and all that. Certainly for me, coming from the University of British Columbia, I don't think there's anyone else in British Columbia who's doing this, at least like getting them beyond like just a couple centimeters in length. And as far as these concerns about like research permitting or like a biosecurity perspective, again, I unfortunately am not very familiar with things too much on the US side, but here I recently just had went to a little local symposium with that had some people from the local BC government, and it seemed like they definitely were just like struggling to catch up with like how fast things are moving in the kelp field right now. They were just currently like trying to get a better understanding of like what people are even doing, or kind of like what's out there, or how things are connected in the wild. So I don't really think there's a lot of like a framework out there right now as to like involving closed systems or any kind of like this biosecurity aspect yet.
Thank you so much. I think we're going to take one last question before we need to move on to the final presentation. Filipe, did you have a question?
Sorry, no, yeah,
Okay.
my hand was still up. Yeah.
All right. Well, thank you again, Varoon. Fantastic. Lots to think about and digest there.
Thank you very much for the invite. I hope it was interesting.
Yeah. We'll be moving on to Brooke, who I saw on, and also Robin, I believe is on. Brooke is from Western Washington University / UW, and Robin is here from the University of California, Irvine. And I think you'll be tag teaming the presentation here a little bit, talking about a recent publication around the interactive effects of temperature and nitrogen on the physiology of kelps. So, Brooke, Robin, take it away.
Yeah, thanks so much. And I think I'll be primarily presenting because Robin was supposed to be in the field today, and then things happened. So, but maybe we'll do questions together. It's the last presentation and we get Zoom fatigue. So why don't we all wake up a little, do a kelp dance, everybody. Woo! Wake up your body. Okay, so
And just a reminder, this one is a little shorter presentation, around 10 minutes. I think you're going to do a power talk. All right, great. Thank you.
Great. Okay. Can everybody see my slide in presenter view and hear me? Okay.
Looks great.
Awesome. Great. Okay. So, yep, I'm going to be talking about very recent publication on the interactive effects of temperature and nitrogen on the physiology of two different kelps. So this was a very collaborative project. It came out of my postdoctoral work at Friday Harbor Labs and was one of also Robin Fail's PhD chapters. And Robin is now a postdoc at UC Irvine working with Matt Bracken on more kelp physiology. And this was just such a wonderful collaborative effort. And so I'm excited to talk to you about it. I would like to acknowledge our funding sources. This was largely funded by the Washington State Legislature proviso for kelp conservation in 2021 to 2023. And we also got some funding through Robin's grant with the Northwest Climate Adaptation Science Center. And I'd like to acknowledge our co-authors, Helen, Megan, and Emily, as well as the team of folks who helped support this research at Friday Harbor. Okay, so we all are familiar. We're working up here in the Salish Sea of Washington, where bull kelp is the dominant canopy-forming kelp species. But as we all know, we've had substantial bull kelp forest declines in Central and South Puget Sound particularly, and we've lost about 60 to 80% of bull kelp in South Puget Sound. We know from the work of Helen Berry and others that bull kelp forests are declining in warm and nutrient-poor regions in Washington, particularly in South Puget Sound. So I'm sure that some of you are familiar with this graph, but here is a zoomed-in version of South Puget Sound. And you can see the historical presence of bull kelp is indicated in this blue color. So those are places where we used to have kelp forests in South Puget Sound, and then these two pink places are the only remaining populations. And these declines have been associated with particularly warm summer water temperatures combined with low dissolved organic nitrogen levels.
Just had to mute the audio and the thing that I had running.
Okay, we're good. So it's possible that this combination of warm temperatures and low nitrogen levels is the result of stratification. So you get a warmer water mass that gets depleted in nutrients from phytoplankton. So we have these declines associated with high temperatures and low nitrogen concentrations, but in the marine environment, temperature and nitrogen are often correlated stressors. On the outer coasts with upwelling, you get cold, nutrient-rich water. And with the lack of upwelling or stratification, you get warm, nutrient-poor water. So our kelp are declining in some of these regions that are warm and nutrient-poor, but it's really hard to separate out these stressors because they occur simultaneously. And so in order to tease them apart, we did lab experiments where we could vary both temperature and nutrient levels to see how they interact. And it's also really important to look at nutrient levels, especially because with climate change, future increases in upper ocean stratification will reduce nutrient and nitrate levels. And some of the largest declines are predicted to occur in upwelling zones. So we do have a general trend of decreasing upper ocean nitrogen as well as increasing temperatures. And there's some hypotheses in the literature about nitrogen. And basically, studies have found that it may increase the thermal tolerance. This study with blades of Macrocystis found that over short-term incubations, adding nitrogen increased their photosynthetic rates in warmer temperatures. So it's possible that because kelp requires so much nitrogen to grow, adding nitrogen might increase their resilience to these warming stressors. And so we wanted to tease apart the effect of temperature and nitrogen on adult kelp sporophytes in the Salish Sea. So Robin and I together conducted two different experiments, which I'll tell you about now. So the first experiment looked at how thermal stress and nutrient depletion affected bull kelp, Nereocystis, across two different populations. So we collected adult sporophyte blades from Turn Rock, which is right near Friday Harbor Labs, and it's a very cold water site, and Cherry Point, which is over north of Bellingham, and it's close to the Fraser River outflow. And you can see from this temperature data, I put out temperature sensors over a year, and Turn Rock remained relatively cool, but Cherry Point got very warm in the summer. The top graph is daily mean temperatures, and the bottom graph is the daily maximum temperature. And you can see that Cherry Point was reaching 16 and even 18 and 20 degrees. And this is on the bottom at -3 to 5 meters. So these temperatures are getting really warm even at the bottom of these kelp forests. And we wanted to see if the population from the warm water site responded differently to these stressors than population from the cold water site. And perhaps the warm water site was more adapted and had greater resilience to warm temperatures. And then in a second experiment, we looked at a comparison between the bull kelp Nereocystis and sugar kelp Saccharina. The reason is because in some of these places in southern Puget Sound where we've lost bull kelp, Saccharina persists at the bottom. And so we hypothesized that Saccharina had a higher physiological tolerance to these warm and nutrient-poor conditions than Nereocystis, given its persistence in some of these declining sites. And to do these experiments, we collected blades of these species out in the field. Bull kelp, we just collected them from the surface, and we went scuba diving to collect some Saccharina from the bottom. We brought them into the lab, this temperature-controlled lab at Friday Harbor Labs with these large tanks. We had 3 different temperatures, 13, 16, and 21 degrees. And then within each temperature, we maintained a low nitrogen treatment and a high nitrogen treatment. I will say that the second experiment comparing the species, our low temperature was 9 degrees because Saccharina is on the bottom. So we had that experiment have a slightly lower cold temperature. And then we slowly increased the temperature over 5 days of an acclimation period. And then we had 8 days of full treatment periods at these temperature and nutrient levels. So just to see what that looks like, we put the blades into the tanks and we actually let them naturally draw down the nutrients, and the nutrients became relatively depleted in about 24 to 48 hours. And then we targeted daily nutrient additions to try to hit these nutrient levels of less than 5 micromolars, which represents those surface stratified nitrogen depleted waters. And then we aimed for about 30 micromolars in the high nitrogen treatment to simulate like a good upwelling day. So we had that 5-day acclimation period, increasing the temperatures, and then we had these 8-day full treatments. And we looked at change in biomass and other measures that I'll talk about in a minute over that treatment period. And it was really hard to calculate exactly how much nitrogen we needed to add and to predict exactly how much we needed to add every day to maintain those treatments. So in the first experiment, we overshot a little bit, and our high nitrogen treatment started around 25 micromolars, and at the very end had reached 100, which is very high, with a mean of about 80. But the low end treatment remained below 3 micromolars. So we did have that really nice difference. And then we adjusted our nitrogen levels and really hit it on the spot for the second experiment with the species comparison. And our high nitrogen treatment was like 10 to 20 micromolars, and our low nitrogen treatment remained around 1 micromolar of nitrogen. We looked at numerous physiological response variables to get a holistic picture of how these stressors are impacting these kelp blades. We looked at the ratio of carbon to nitrogen in their tissues, and I'll just tell you that the high nitrogen treatment had a lower C to N ratio, indicating nitrogen enrichment. So in both experiments, the C to N ratios were significantly different. So that's just an indicator that our nutrient manipulations worked. We measured multiple aspects of growth, but I'll just show you change in biomass here. We measured photosynthesis and respiration rates using an oxygen production method. We did PAM fluorometry to look at fluorescence, but I won't show you that data because it largely didn't differ across treatments. We quantified nutrient uptake rates of nitrate and ammonium. And then we also did a collaboration with Kathy Pfister's lab looking at microbial communities. So I'm going to show you a series of graphs, and I'll just orient you. These graphs are the left will be from that 2 population experiment. The Cherry Point kelp are in red and the Turnrock kelp are in blue. And nutrients, the solid ones are high nitrogen and the open ones are low nitrogen. So this just shows the change in wet weight or biomass, and 0 indicates that there was no change. And so you can see at the 2 colder temperatures, there was a positive increase in biomass. The blades grew like 8 to 10 centimeters over the course of the experiment. So we saw that in the cold treatments, they were really happy and they were actively elongating. But at 21 degrees, both populations had about 0 change in biomass. So they did not grow and they weren't doing very well. And in the multiple species experiment, again, we found that at the cold temperatures, both species increased their biomass. But in this experiment at 21 degrees, both species responded very negatively and actually lost biomass. So together, this shows that across both populations and species, 21 degrees was a really critical temperature where they either didn't grow or they lost biomass. And that happened regardless of nitrogen levels. So we didn't see that adding nitrogen had a positive effect on growth, even at the warmer temperature. Basically, temperature was the main stressor here, and nitrogen didn't really help them out. And you can see this, these are just representative photos of Nereocystis from the 2 populations, as well as the 2 species. And you can see that at 21 degrees, the blades physically deteriorated. They started dissolving, and this was after only 8 days of exposure to these high temperatures. And what's really fascinating is you can see that they physically deteriorated regardless of nitrogen levels. And this is something that we've observed in the field at some of the warmest sites in British Columbia and in Washington. We've seen that these blades have physically deteriorated, and we're wondering whether it's from nutrients or from temperature. And this indicates that it's probably mostly just a temperature effect because it happened across nutrient levels. We measured respiration rates in the dark, and we found, so here it's a little confusing because respiration removes oxygen from the water. So more negative equals more respiration. And so we found that basically respiration rates increased significantly at 21 degrees across both populations and species, and that's indicative of stress. You're respiring more in these warmer temperatures. We did see that net photosynthesis rates either increased or showed a hump-shaped relationship with temperature, which is really interesting. We thought that because we had such low growth rates that we would actually have seen a decline in net photosynthesis. But what this just means, I think, is that even though they might be photosynthesizing more at higher temperatures because it's an enzymatic activity that's controlled by temperature, that was not enough to compensate for all the cellular damage that occurred at those high temperatures. So even though they might still be fixing carbon, they're incurring a lot of damage and have to repair that damage in their tissues. When we looked at nutrient uptake rates, we found, unsurprisingly, that in the high nitrogen treatments, they take up more nitrogen because it's available, and kelp, they respond to increasing nutrient levels by taking up more nitrogen from the seawater. But what was really interesting is that we did see an increase with temperature. We weren't able to measure the warmest temperature in the species experiment because those blades were so degraded. But we do see this increase in nutrient uptake with temperature, but in Puget Sound, obviously, that will only benefit the kelp if there's still more nitrogen available to take up. So you see in those low nitrogen treatments, they just didn't increase because there wasn't that much more nitrogen to take up from the water. And then we also collaborated with Nikos Molnar, who's an undergraduate at University of Chicago, in Kathy Pfister's lab. And we swabbed these kelp before and after the treatment period to look at how microbial communities might have shifted. And we found the greatest microbial community shifts associated with the highest temperature. And at 21 degrees, the microbial diversity actually increased as microbial taxa invaded from the seawater. So there's potentially functional implications associated with shifts in the microbiome. So to conclude, we found that high temperatures were stressful for both species and populations, and they increased their respiration rates and had physical deterioration of the blades. Low nitrogen was surprisingly not stressful, and additional nitrogen did not mitigate that high temperature stress. In our comparison of two different species, Saccharina did not outperform Nereocystis, so both species were equally susceptible to the effects of warming. And we like to conclude that nutrient additions are unlikely to enhance these the physiological performance of kelp at these high temperatures. I've presented this work before, but I also looked at the effects of temperature and nitrogen on these juvenile sporophytes and those microscopic gametophytes. So you can look at that paper. But those had a lower temperature limit after which they died. And we found in this paper that 21 degrees was a critical threshold for the adult sporophyte blades. And with that, I'll take any questions. And Robin is around too.
Thank you so much. Really fascinating. All right, folks. So if you have your questions, feel free to raise your hand or put them in the chat. I know Helen always has a question or two. Speaking
Yeah,
of.
I'm happy to start this off. This work is fascinating to me because I think many of us who work in kelp have a one of the fundamental rules is that kelp responds positively to high nutrients, and temperature is a proxy for in much of the kelp research we read, temperature is used as a proxy for nutrients. And so for me, this is a really interesting and surprising and also somewhat subtle result that makes me think about decoupling all of my assumptions about temperature and nutrients. And also wondering if survival in Puget Sound, a place like Salish Sea, where high temperatures are come with low nutrients, maybe that combination facilitates greater survival, which is very counterintuitive to my expectations. And I'm wondering if any other people are struck by this and have other comments, along with Brooke and Robin. We've been talking about this a lot. It's a very counterintuitive result, and we've gotten it multiple times.
Robin, do you have anything to add about that?
I think we've talked about this a lot, and it is a surprising result. And yes, I do agree that it is interesting that temperature has been used as a proxy for nutrients when I think that we should be viewing them more as discrete stressors, and we should be considering the nutrient data separately and more explicitly than it has been done in the past.
I'll just address a question in the chat about C to N. So we did measure the C to N ratios from like the natural populations that we collected in the field, and then we measured them. So those were like the initial condition, and we measured them again at the end of the experiment. And we found that in the high-end treatments, the C to N ratio decreased from the initial. So we did find evidence that adding nitrogen was incorporated into their tissues and led to a change in the C to N ratio, whereas the low nitrogen kelp didn't have that decrease compared to the field collected samples. Let's see. And there's another question about persistence of prostrate sugar kelp in areas where bull kelp has declined and the slightly cooler subsurface temperatures there. And yes, I think that's a really great question, because we didn't see a difference between the two species in response to these stressors, yet Saccharina persists in places where bull kelp has been lost. And we hypothesize that that's because it has a colder water refuge on the bottom. Like they might be experiencing colder temperatures because they're laying along the benthic zone, whereas bull kelp is up in the surface in that really stratified warm water. But again, I want to point out that our temperature data also show that it is reaching 16 or 18 degrees on the bottom, but not really reaching 21 degrees where we found the stressful effects on the sporophytes. So I do think it's interesting to think about colder water refuges for understory kelps persisting in areas where the bull kelp might be really stressed floating in the surface.
And Margot's got a question. Margot?
Yeah. It was, well, kind of not so much of a question, but just building on Philippe's comment, Filipe, is that perhaps the tight coupling is based on a lot of work from California where there is that strong environmental coupling because of upwelling. And so when we look at our data from British Columbia and pair it with kelp growth, bull kelp growth parameters, we also see some decoupled relationships throughout the season, with nutrient limitations likely at times when they're lower, like in the early growing seasons, and then temperature playing more of a role in the summer. So probably fairly similar to Puget Sound.
That's really interesting. So like how low is your nitrogen concentrations getting, and you still see positive growth rates and things like that?
Yeah, so it's, I don't know the exact, and this is on Dean's work, so she knows it in a lot more detail, but yeah, so down to some of the like limitation levels, like below, what is it, 0.001 or 0.01 micromolars. Yeah.
Wow. Yeah. Yeah, I mean, it sort of seems like kelp can grow at these low nitrogen concentrations. Something Robin and I and Helen and everybody has talked about is like, even though nitrogen is low, if you have flow, you have a constant delivery of nitrogen, even at that low level, that might be enough for them to take up and persist.
Yeah, and that also addresses one of the questions was like, how might current? So like, I think that the current and flow is really an interesting avenue that should be pursued further in the kelp work.
Thank you so much. We do have time for one more question from Ron.
Hey.
Oh, thank you. Yeah. Can you hear me? Okay. Yeah. So it's a great talk, and what's emerging with me is a threshold of temperature. We've done a bunch of work on the physiology of eelgrass, and it looks like about 16, 17 degrees, the respiration rate really starts to go up, and net production goes down. And it's kind of around the same numbers that others are getting, including you, about, well, you got between 15 and 20, but you know, there's something to be said, especially with these anomalous weather events like in 2016, where we have this blob that things really heat up, and that might be the period where they're really vulnerable. But great talk. Thank you.
Yes, thanks again. Great discussion and good stuff to think about here as we move into our roundtable. So we just have this last 10 minutes. It's a little more informal. And there may, Brooke and Robin, a couple questions may continue to come into the chat. You feel free to address those. This roundtable is more informal, but we do want to start with allowing some space for Andrea Jackman. Are you on?
Yeah, hi, I'm here.
Yeah.
Yeah, it was really great to hear all the talks today. My name's Andrea. I'm a fourth year undergrad, and I'm doing my honors thesis with Laura Parfrey at the University of British Columbia. I've been working with a grad student to look at red spot disease in sugar kelp because it's been a problem for some aquaculture companies around here. We still have a lot of unanswered questions about disease. So we developed a survey, and we would really appreciate it if we could get some input from people working in aquaculture on disease. The link is in the agenda, and I'll also just post it in the chat right now. Let me just grab that link. There it is. So this is going to contribute to my thesis and also help us develop future work investigating disease in the lab. So the first few questions do ask about your name and email, but if you want to remain anonymous, you don't have to fill these out. We're basically just asking to be able to get in contact with you if you're interested in that. And any personal information will be separated from the data before we share it. I was hoping that everyone could take a few minutes right now and just fill it out. Please let me know if you have any questions about it. I'll also put my email in the chat if anyone wants to reach out later. But yeah, thank you so much for giving me a couple minutes to share this. And thank you to anyone who fills out the survey.
I have a question.
Yes.
Do you want us to just fill it out if we're involved in aquaculture? What if we just do research?
Yeah, if you're involved in aquaculture, if you've encountered disease, we really appreciate any information. So it'd be great if you could fill it out.
Okay. I haven't encountered disease, so I'm just wondering if you want like negative data in addition to,
Yeah.
I mean, I'm not aware of it from anything we've outplanted, but.
Yeah, I definitely do want also negative data would be great, just so we can try and identify any patterns.
Great. Hillary, I saw you came off on video. Did you have something you want to share at the roundtable?
No, thank you. Just showing up for roundtable-ness.
We appreciate that. It's great to see your face here. Greg, you're showing up too. Anyone else? Okay, we have a hand. Jeff?
Yeah, I just wanted to let people know of a document that is currently out there that hopefully will be helpful to you all. As a lot of you know, there have been a lot of new efforts popping up in the kelp world, specifically various plans, strategic actions. And so there's a large number of us that came together to try and decrease confusion about who's doing what's going on. And we created a document that is kind of a comparison document that features a number of the large ongoing collaborative strategic plans initiatives, as well as work groups. And so I just want to let you know that is available. I'll just really quickly, I'm going to share my screen if that's allowed, just to show you what it looks like, and I'll show you where it lives too, if you're interested. This is it. It's 4 pages long, but it's just basically highlights. I can't remember how many we have in here, 8 or 10. Looks like the kelp plan, the health plan, the kelp node, and again, a lot of those other efforts. Has a general description, links to each comparison guide, kind of like when you go on the internet, you want to shop at, compare the different things there, whether vacuum cleaners or whatnot. This is that. And then it also has a relationship table showing how they're all connected together, with the largest connection being information sharing, which is this kind of peachy yellow square. So I just want to let you know that's there to help guide you through and hopefully decrease any confusion about all these different meetings that you're going to. So I'll put the link in the chat. So thank you.
Thanks, Jeff. Trying to bring some clarity to spaghetti, all the networks. All right. We have just about a couple more minutes. Is there any last burning roundtable updates folks want to get out before our next meeting? All right. Helen, you want to give folks an update on our next meeting? Anything we should know about that, when the date is?
Thanks for coming, everyone. Our next meeting is May 15th. We look forward to seeing you, and we will post a recording of this meeting. And with that, we'll give you 4 minutes back to your lives. It's 11:56. Take care, everyone. It's a pleasure to be in the same work group with all of you.
Thanks, everyone. Thanks.